Hi, welcome to the Bioinformatics Chat. I'm Roman Jipliaka, and today I'm talking to Mingfu Shao. Hi, Mingfu.
Hi, Roman. Hi, everyone.
Mingfu is a Lane Fellow at Carnegie Mellon University, and I recently read his preprint that he and Carl Kingsford published on bioRxiv that describes his software called Scallop. And Scallop is a transcriptome assembler. Mingfu, I want to talk to you about Scallop today, but before that, let's get to know you. Could you tell us a bit about your background?
Yeah. So my background is in computer science. So my bachelor was in Beijing Institute of Technology, and my master is in Chinese Academy of Sciences. So during master, I worked on something related with protein folding and protein structure prediction. And then I went to EPFL called Swiss Federal Institute of Technology in Lausanne, Switzerland, where I worked on comparative genomics, especially in designing algorithms to solve those edit distance problem under genome rearrangements. So after that, I joined CMU as a Lane Fellow working with Carl Kingsford. And my project is focusing on transcript assembly.
Very impressive. That's a very diverse set of bioinformatics problems. What motivates you to work on these problems?
I'm interested in algorithm design, and also I'm interested in solving real world applications. So not just Q theoretical thing. So I think in bioinformatics field there are many important problems that requires algorithm design. So I think Scallop is a very nice example to illustrate the research that I'm interested in. To solve the transcript assembly, you need to first formalize that as a mathematical optimization problem, and then you need to figure out why it's hard, what's the property, and what's the insights, and then you need to design an algorithm according to those insights. So after that, this is the theoretical part. So after that, I'm also interested in implementing the algorithm to provide a tool or software for users.
And were you familiar with transcriptomics before you came to Carl's lab?
No, it's a completely new small field for me. So I never touched, the sequencing technology, the reads. I never work on this field. So transcript assembly is a new problem for me. So I found it's interesting because it's still open and challenging. So there are many existing assemblers, but they are still not accurate enough. So there is big room to improve them. That's one reason. The other is also this problem is also very algorithm interesting. Yeah, so I think those two factors are the reason that I choose this problem to work on.
Cool. So do you plan to continue to work on this problem in the future, or would you like to try something else?
I think I will keep working on transcriptomics and also maybe cancer transcriptomics. So Carl's group has pretty diverse topics, so I might collaborating with my colleagues on working on cancer. So particularly my interest in is solve those computational challenging problems and provide tools and softwares for our community.
Okay, so let's talk about the community and about some ways in which the community could use a tool like Scallop. And perhaps we should start by explaining what RNA-seq is, just in case any of our listeners are not familiar with this. And then you could explain how Scallop helps to analyze the RNA-seq data.
So RNA-seq is a technology that can be used to capture the expressed transcripts in a sample. But we usually got the short reads, not the full-length transcripts. So we need to reconstruct the full-length expressed transcripts from the sequencing reads. So this is the task of transcript assembly. And Scallop is a reference-based transcript assembler, meaning that you require a reference genome. So, and before assembling, you first need to map all the sequencing reads to this reference genome, and you got the reads alignment. So, as a software, so the input of Scallop is just the reads alignments in BAM or SAM format, which can be generated by some RNA-seq aligner like top hat, star, and high set. And the output of Scallop is the predicted expressed transcripts in GTF format.
So not everyone has to assemble their own transcriptome, right? So for example, if people work on a model organism, well, like a human, they already have a high-quality pre-assembled transcriptome. So for these people, is there any reason, perhaps, to assemble their own transcriptome as opposed to using a pre-assembled one?
Right. I think that's a very great question. Scallop gives like sample-specific transcriptome. So it only reports the expressed transcripts in a given sample. In a sample, only 20 or 30% of the whole transcriptome will be expressed. So if you only use the whole transcriptome, then it's clearly a like overestimation.
But presumably a good quantification tool like RSAM would recognize that these transcripts are not in fact expressed and just assign zero weights to these transcripts. So is there any reason to want a smaller transcriptome?
Yeah. So I think one reason is that the current transcriptome is not perfect. There are like many evidence saying that many transcripts are still remain to be annotated. So in terms of this, Scallop can be used to like identify novel isoforms which are not in the current transcriptome.
Right. So your main selling point must be that you're able to find those rare transcripts that are not present in the reference, whereas being able to filter out the transcripts that are in the reference but not in the sample, that may be a bit less important, right?
Yeah, right. Yeah, right. And in terms of this, it's especially useful to apply Scallop on those spaces that don't have a good annotation database.
My impression has been that whenever you assemble something, be it a genome or transcriptome, you generally need a pretty deep coverage. Does Scallop work well under low coverage?
Yeah, I think when you have lower coverage, it's better to use a reference-based transcriptome assembler. Because when you do a reference-based assembly, the most important thing is to correctly identify those junctions. So even if the coverage inside exon is very small, it doesn't matter because you use reference-based. The reference genome can help you to bridge the intron, as long as the junction can be like mapped or covered by enough reads. So that's why usually a reference-based assembler is more accurate than a like de novo because it allows very low coverage inside intron exon.
Yeah, I appreciate that Scallop or another reference-based transcriptome assembler can work better under low coverage than a de novo transcriptome assembler. On the other hand, if you compare this to just using a existing transcriptome, is there any reason to use Scallop if you have low coverage?
You can try another thing, like you first apply Scallop to assemble the transcripts, and then you merge those expressed ones with the known transcriptome database. So now you have a bigger set of transcripts, and then you apply RSAM or other quantification tools to quantify them.
That's a good point. So as we discussed earlier, if you have a good quantification algorithm, then having a few extra transcripts, it shouldn't hurt, right?
Right, yes.
I recently saw a talk by Ben Langmead, and I will put a link to the talk, to the video and the paper on our website, bioinformatics.chat. So I saw this talk, I watched this talk, and Ben describes his work. It's called Rail RNA, and it is a spliced aligner for RNA-seq reads, which performs well on a huge amounts of RNA-seq reads. And they aligned a lot of reads from the SRA to discover novel junctions. And in order to save disk space and some time as well, they don't typically output the BAM file. They don't output the aligned reads themselves, but they only output the junctions that they found. So I'm curious if Scallop could only read those junctions. If it could not require a BAM file, then Scallop could be used with Rail RNA to assemble a transcriptome without any alignments.
I think I only need the junctions and also some additional information about the cigar. So how a read is aligned to the reference genome.
Ah, okay. And what do you need that information for?
I think it's very important because one read might span multiple exons. So if you look at their scar, it's some match and then some N. N means a junction and then some matching and then some junction. So that means a read can be mapped to multiple exons, which is very useful to provide, we call them phasing information to reconstruct the transcripts.
I see. So you require that phasing information in addition to raw alignments.
Yeah.
But presumably there is a more efficient way to store and perhaps aggregate the phasing information than a BAM file. So maybe Ben could alter his software Rail RNA to output this phasing information in some new format. And you could alter Scallop to read this phasing information. And that would make a pretty cool tandem because you could assemble a better human transcriptome based on that enormous amount of data.
Exactly. Yeah. I don't use the reads stored in the BAM file. So I use BAM file because it's convenient. It's like a community standard. Yeah, as you said, the junction and phasing information are the only thing I need.
When you worked on Scallop, what was your development process like? Because Scallop is both an algorithm and an implementation. So did you design the algorithm first and implemented it later? Or did you work iteratively and develop them simultaneously?
At the beginning of this project, we focus on algorithm, focus on theoretical part. So I think that takes about half a year. We came up with some theoretical results, which is called minimum path flow decomposition problem. And we came up with the algorithm for that. But that problem is a bit different from real case of transcript assembly. And then after that, we try to, working on the real datasets to see whether this theoretical model can be applied to real datasets. And we found that there's some problem there. And then we switched to design more specific algorithm for more realistic scenarios, transcript assembly.
Yeah, I think it's a great approach when you first start with a simpler problem and see if you can at least solve that first. So let's discuss that mathematical model that you came up with.
Maybe we start from an example. For example, we have a gene with multiple exons. And also this gene has multiple spliced isoforms that are expressed. Thinking about the situation that two expressed transcripts share a junction, then we only observe the superimposed abundance of this junction instead of the individual abundance of the two transcripts that share this junction.
So to clarify, the abundance of a junction is basically a number of reads such that the first half of the read maps to the first axon and the second half of the read maps to the second axon. And this is a junction between the two axons. So you count the total number of reads that map in this way. And this is your junction abundance, right?
Exactly. Yeah. So the situation is we only observe this merged thing. So it's a graph, like each axon is a vertex and each junction is an edge. So what we observe is this merged graph from many like individual transcripts. So the problem is to reconstruct those individual paths from this like merged graph. We call it splice graph. If we further assume that each transcript contributes equally to the junctions that it has, then this problem can be formulated as a flow decomposition problem. If we further assume the parsimony assumption, like we want to explain the splice graph with minimized number of paths, then this becomes a pure mathematical problem called minimum path flow decomposition problem.
And perhaps this is a good time to explain what a flow is.
Yeah. So a flow of a graph is defined on the weights of the edges. So suppose that there's a weight for every edge. So what is flow? So a flow just need to satisfy this condition that for each vertex, the sum of the inflow should equal to the sum of the outflow. Let me repeat. So for each vertex, the sum of the weights of the in edges should be identical to the sum of the weights of the out edges. So if that condition satisfies for every vertex, then we say that, okay, the flow on the edges form a flow.
And the splice graph automatically satisfies that property because each read covering a junction contributes equally to the left axon and to the right axon of a junction, right?
No, I think the reason why we assume that it's a flow is that it's merged from many paths. But it's very important that in reality, this is not a flow, because in reality, for each transcript, so the reads for different junctions are different, might be different. Of course, it should close to its real abundance of this transcript. But, sequencing process is a random sampling process. It might end up with more samplings, more reads on this junction, but less reads on the other junction. So this makes that in real situation, it's not a flow.
I see. So I was imagining perhaps a different graph, a dual graph to the splice graph where the junctions are vertices and axons between junctions are edges. But the splice graph is the one where axons are vertices and junctions are edges, and the weights are attached to junctions. So this graph will be a flow, for example, if the coverage is precisely uniform across all transcripts.
Right, yeah. That's actually an assumption, which is not the case in reality.
All right, so you make this assumption. You assume that your splice graph is a flow. What next? Were you able to decompose the graph into transcripts based on this assumption?
So this problem is NP hard, meaning there's no efficient algorithm until P equals to NP. So there are existing some heuristics, and we did something more. And our main contribution is some insights about this problem. So we proved some property. We proved that the gap between the number of optimal paths and a known upper bound is determined by the number of independent vectors in some space. So this very important insights actually gives us iterative algorithm. So we can try to identify such vector and then use the vector to reduce the gap. Actually, it's a gap reducing algorithm. So to make our solution as close to the optimal solution as possible. Right. That's the main idea we proposed in the flow decomposition paper.
So you found this efficient, good heuristic for this theoretical problem, and then you try to apply the same heuristic to the real-world problem of transcript assembly. Was that easy? Did it apply well?
So it's related, but also has two difference. The first is I just mentioned that in transcript assembly, it's not a flow anymore. Although the information is still there, but you can imagine it's a flow with errors, with noise. The other difference is in transcript assembly, we have additional information of phasing information or phasing paths. So that are those reads that span more than 3 exons. So in real transcript assembly, we have those two differences. Actually, we design a pretty different algorithm with flow decomposition. Right. So the main idea here is to iteratively decompose the graph. It's still decomposed, but we guarantee that when we decompose the graph, we preserve all the given phasing paths. So this is one clear difference from the flow decomposition paper. Also, when we decompose the graph into paths, we divide all the vertices into 3 types, namely trivial vertices, and non-trivial vertices, and for non-trivial vertices, we further divide them into splittable vertices and unsplittable vertices. And we define different subroutines to decompose these 3 types of vertices.
Yeah, let's dive deeper into the algorithm now. Let's talk a little bit about these different types of vertices. And to remind our listeners, the vertices correspond to axons. So what are the trivial vertices, for instance?
So vertices correspond to axons. By trivial vertex, I mean the in degree of this vertex is 1, or the outer degree of this vertex is 1.
So if I understand correctly, you call a vertex trivial if the corresponding axon is in all transcripts in which it participates, it is always to the right of the same axon or to the left of the same axon.
Right.
Or perhaps it is always the first axon in the transcript, or it is always the last axon in the transcript. Does that also count?
That also counts, yeah. I think that's a very good summary, yeah.
Okay, so how does that help you knowing that a particular axon or a particular vertex is trivial? What do you do with it?
For trivial vertex or trivial axons, there is only unique way to decompose that. So maybe let's take the example axon A is trivial. For example, there's only one axon, which is B, that points to A. And there are 2 other axons C and D, which points from A. So it's B, A, C, D. B points to A points to C, and also A points to D. Then we can decompose this part as follows, B, A, C, and B, A, D. So A will be splitted into 2, right?
Yeah, so decomposing a vertex means that you split it into 2 vertices and you split the original edges of that vertex between the 2 new vertices. Is that right?
Exactly. Yeah. Yeah, for this case, it's clear that there's only one explanation what's happening. So it's 2 transcripts. One goes from B, A, C, the other goes from B, A, D. Yeah. So there is no ambiguity. Yeah.
Okay, so what about the other types of vertices?
Right. So for other types, it's it's a bit complicated. So because for each vertex, we see it's non-trivial, meaning the input junctions are more than 2, or is more than 1, at least 2. And also the output junctions are also at least 2. So that, end up with multiple possible decomposition. So the situation is not unique anymore. So for this non-trivial vertex, we further divide them into 2 types, depending on the phasing information. So if intuitively, if the phasing information covers all the possible junctions related with this vertex, then we say it's like unsplittable. Meaning that, okay, we can decompose this axon completely depending on the phasing information. Like the phasing information is enough for us to decompose this axon. So we call it unsplittable vertex.
It's a bit paradoxical because it sounds like for unsplittable vertices, you actually know precisely how to split, how to decompose them, right?
Yeah. Yeah. So, it is. Yeah, you're right. So it's a bit, it's a bit not intuitive in terms of the name. So maybe we compare with like splittable vertex. Yeah, I think that's easier, right? Splittable vertex means that we can split this vertex into two to try to like minimize the number of paths required. So because for splittable vertex, we don't have enough information. We don't have enough phasing information. So in that case, we try to apply the parsimony assumption, try to reduce the number of paths. So how to do that? We try to split this vertex into two. That helps us reduce the number of paths required.
So splittable vertices are those for which there is not enough phasing information available, and the problem becomes under constraint, and then you have to impose this additional parsimony principle.
Right. Maybe let me give an example. Let's consider one axon, like axon A, with two input junctions and two output junctions. So for example, with the two input axons, one is covered by 10 reads, and the other junction is by 100 reads. And for the two output junctions, one is covered by 10 reads, and the other is covered by 100 reads. So you say the two input junctions, 10 and 100, and the two output junctions, also 10 and 100. Then you would say, okay, one transcript must goes from 10 axon A and then 10 junction. So the other transcript must go through 100 junction and then axon A and those and then this junction with 100 reads. Right.
Oh, that's a great example, and I think it really helps to visualize what's going on here. But I'm curious, what if it's the same picture as you draw, except all four numbers equal 100? So in this case, there is a non-parsimonious answer, which is to have 4 transcripts, each with the abundance of maybe 50. Or we could apply the parsimony principle, but then we have to split the 4 junctions among 2 transcripts in a arbitrary way. So we can explain this picture with just 2 transcripts, but there are 2 ways to explain this picture with 2 transcripts. And of course, only one of them may be correct, or maybe neither. But if you make this decision to assume only 2 transcripts, then the pair you have to pick is pretty arbitrary, isn't it?
Yeah, you're right. So if there's no phasing information, there's no such covered information to help us divide it, then we have no information to do. So I believe the result is like kind of arbitrary. So if like 2 input junctions are both 100 and 2 output junctions are also 100, then no information to tell us how to go.
So in order to prevent this from happening, I think the answer is just have more phasing information, right? And if we have enough phasing information, then all vertices become either trivial or unsplittable. And what are the ways in which we can supply more phasing information to Scallop?
Yeah, I think that depends on the data we have. I think the longer reads we have, the more powerful phasing information we have, and then we can expect more accurate results. Yeah. Right. So I think the length of the reads is one important factor, and also the coverage. The higher coverage we have, the more accurate results we can expect. Because as we just discussed, for those vertices, for those splittable vertices that are not covered by enough phasing information, we rely on the coverage information. So yeah, so the higher coverage helps us decomposing splittable vertices. Yeah.
So would it be ideal for Scallop to use reads from something like PacBio or Nanopore, right? Those sequencers produce very long reads, and very long reads give you phasing information.
Yeah, this is a very good question. So actually, in the current submission, we haven't done that. We haven't applied it to like PacBio long reads, but so those long reads can be like easily incorporated into our framework. So just model them as like phasing paths. So the algorithm we provide is a general framework that can be applied to reads with any length, and we guarantee that all phasing paths information can be used.
I also found it interesting that you use linear programming in Scallop. Could you talk a little bit about where linear programming comes up?
Yeah. So the linear programming is used to decompose unsplittable vertices. So as we discussed before, so what is unsplittable vertices are covered by enough phasing information. So then we try to decompose this vertex according to the phasing information, but we also want to assign the weights to minimize the deviation from the observed coverage. So here we have two objectives. One is to try to minimize the number of paths required. The other is to try to minimize the deviation from the observed read coverage. But for unsplittable vertices, the number of paths required is kind of fixed because it's determined by the phasing paths. So then we only have one objective, which is to minimize the deviation from the observed coverage. So in this case, we use linear programming to minimize the deviation from the read coverage with the constraints that it follows exactly the phasing information tells us.
Here's what I find confusing. So we said before that unsplittable vertices can be decomposed in a unique way. So if that decomposition is unique, why do you need to optimize anything?
Because you know how to it goes, but you need to assign the new weights for each decomposed path. For example, again, like we have two in junctions and two out junctions. And after decomposition, this vertex will disappear, and we end up with some new paths. But what's the new weights for these paths or these new edges? This should be determined here by making them as close to the observed coverage as possible.
Okay, and you said you only do this for unsplittable vertices. But doesn't the same problem come up for splittable vertices?
With splittable vertices, we only need to determine how to split that. So the edges will keep the same. So there's no need to assign new weights, because after splitting, so some edges will attach to the original vertices. Some edges will be attached to the new vertex. So there's no need to reassign weights. Just move some edges somewhere.
I see. So for splittable vertices, you have a different kind of optimization problem than for unsplittable vertices. So how do you solve that optimization problem?
For splittable vertices, we use the subset sum, we formulate it as a subset sum problem and solve that using a existing dynamic programming algorithm. Because for splittable vertices, so the problem we need to solve is to make them as balanced as possible. Like the example we just use. So we try to, choose a subset of the in junctions and a subset of the out junctions to make them as balanced as possible. So, yeah.
And this is what makes this problem NP hard, right?
Yeah, it's NP hard. But it can be efficiently solved by a pseudo polynomial time algorithm.
So we already discussed long reads or longer reads as one source of phasing information that Scallop can use. But another source of phasing information is paired end reads, right? So could you explain how Scallop uses paired end reads?
Right. Yeah, I think this is also very important. So with paired end reads, so we try to get the phasing information of each mate read, and then we check whether the two mate reads can be connected or can be bridged by a unique path. If the path between them is unique, then we combine those reads together. Otherwise, we don't use the paired end information. Of course, we do use the individual, mate read. Maybe each mate read can still, span more than 3 axons. We do use that information. But if the path between these two mate pair is not unique, we do not use the paired end information anymore.
So in that case, you don't use the pairing information because, while you know that the junctions belong to the same transcript, you don't know which transcript it is. And there may be multiple transcripts. But it seems like if you know the insert size, then you can infer the length of the path, and you can disambiguate between multiple alternative paths based on that length inferred from the insert size, right?
Yeah, right. So I think that's the part we haven't implemented. So yeah, currently we just ignore those paired end information if the path is not unique. But yeah, as you said, we can infer the most likely path according to the insert size distribution.
Is Scallop the first transcript assembler that takes advantage of the phasing information?
So I think some existing assemblers use the phasing information, but some assemblers don't use phasing information. But Scallop can be proved to guarantee preserving all the phasing paths. So that's the most important algorithm contribution of Scallop to this problem. And it is also one of the key reasons why Scallop is better.
But it's quite easy to preserve all phasing information, right? You just report all possible transcripts, all possible junction combinations, like a Cartesian product. So I think the hard problem here is to preserve all phasing information while still reporting a reasonable set of transcripts.
Yeah, right. Yeah, that's true. So in addition to that, yeah, I think that's the one key property we want to have. But we also want to minimize the number of paths required. Yeah, as you said, a trivial solution is just to cover everything and use tons of paths, but we don't do that. We also try to minimize the number of paths under the parsimony assumption. And also, we want to minimize the deviation from the observed coverage. So those three things are combined together in our algorithm.
Yeah, I think it's remarkable that you can balance all these conflicting criteria. And another thing I found cool about Scallop is that you don't only assemble the transcriptome, you, as part of your algorithm, can actually compute the expression of the transcripts, right?
So yes, as you said, it also outputs the expression abundance of each predict transcripts. Yeah. it gives you both the transcript and its expression abundance.
Have you compared the accuracy of quantification performed by Scallop to something like RSAM?
Yeah, very good question. So we haven't compared them. Actually, that's the work I'm doing now. So combine quantification and assembly. So right. We haven't done that.
And in general, how do you evaluate the accuracy of a transcriptome assembler? I imagine it is quite tricky because you don't know the truth.
So currently, and also commonly used method is to compare to the reference, like known database. So for example, for a predicted transcript, we see it's correct if its intron chain can be exactly matched to a known transcript in the database. So of course we know so only a subset of the transcripts in the database are expressed, and there might be some novel ones. But this way is still, very good way to relatively compare the performance of the method. So it's fair for those methods, although it's not, perfect because we don't know the ground truth. Yeah.
Yeah. So if you discover a transcript, a true transcript that doesn't happen to be in the database, then you won't get points for it. You even will get punished for that, right?
Exactly. Yeah, that's true. Yeah, because we realize that the current transcriptome is not perfect, is incomplete. Currently, we are trying to, build a better transcriptome from the large-scale datasets. So we, use many existing RNA-seq datasets and then try to assemble them and try to merge the predicted ones to build a better transcriptome. Yeah, that's also something I'm doing now.
Yeah, I think this overlaps largely with the work of Ben Langmid that I talked about before. So I definitely urge you to look at that because he basically did a lot of that work. He did the spliced alignment of all those RNA-seq reads, and he got the junctions. The only problem is that he may not have the phasing information that you require. And I'm curious if something can be done there.
Yeah.
Well, Mingfu, it was great to talk to you. I think I now understand your paper much better than I did before. And I highly recommend all our listeners to go check out your papers. We will have the links on the website, bioinformatics.chat. And those who deal with RNA-seq data, definitely give Scallop a try and perhaps give Mingfu some feedback. I'm sure he will appreciate it.
Yeah, thank you. I'm very glad to have this opportunity to introduce our work. And so we are open to our users, and we will add features according to our users' feedback and comments. Yeah.
Awesome. This was Mingfu Shao. Thank you for listening.